The first one was go, go. This one is stop. So if they are mutated, there is no stop. There is no break. They are usually loss of function. Both alleles should be mutated. So they are recessive, previous one they are dominant. The third group, which has several names, same as cellular maintenance genes or the genes responsible for detecting mutation and repairing the mutation, again same as tumor suppressor gene, both alleles should be mutated. They are negative regulator and they have loss of function. These two are very similar to each other. The previous one was completely different from the other two. This, sorry? Yeah, you will have, wait. These three groups are very important to know. Proto-oncogene, normal. Oncogene, abnormal, positive regulator. Tumor suppressor gene, negative regulator, both alleles should be mutated, and cellular maintenance gene is the same. At least 350 genes are responsible for cancer genetics. The majority of them are somatic mutated, 20% they are germinal mutated, 10% both germinal and somatic. What does it mean? Germinal, I think I told you before, but I should tell it again because it's very important, somatic. We do have two kinds of mutation. If a mutation is germinal, means a sperm or ovum are mutated, so all the cells has the mutation. Whatever sample you get from urine, from blood, from the buccal smear, you will see the mutation. It means germinal. Somatic mutation is only in the tissue involved. Same as, for example, if you do smoking, you will have lung cancer, and only this mutation is in the lung, not in the stomach, not in the blood, not in the urine. Somatic mutation, germinal mutation. And the gene mutation would be somatic or germinal or both. You will see the example. Another important issue, is it driver or is it passenger? What does it mean? Driver means this gene mutation is responsible for next mutation. So they are driver. Sometimes there is no cause and effect of that mutation. We say it is a passenger. Just seeing the mutation, it is there, but there is no consequence out of it. Driver and passenger should be defined when you find a mutation. And also we say, is it an initiator, or is it progressor, or is it responsible for metastasis? And we know that as well. So the oncogene, when they are mutated, there is no stop on the cell division. There are a lot of proliferation. They have gain of function. On the cellular level, they have dominance. Usually the mutation is in the gene, or is it in the regulatory region, or copy number. You saw this copy number in the cytogenetic as well. Usually oncogenes amplify, so copy number is increasing. But for the tumor suppressor gene, copy number would be deletion, decreasing in the number. Here is a big list of the proto-oncogene. You see the function. You saw about ABL, also BCR, or ERBB2, RAS, RAF, MYC. These are a sequence of several viruses, and we found out on the human being they are responsible to be proto-oncogene. If they are mutated, they will be oncogene, and there will be cancer out of it. You will see the cancer list and the gene behind that. Usually we have signal transduction in the proto-oncogene. Signal transduction means translate extracellular signal into the change in the gene expression. We do have a cell. This is signal from outside, for example, growth factor. We have receptor. It comes to do some change on the nucleus. And we do have several proto-oncogene here working, and you will see them on the red color here. Ret and MET in the signal transduction in the surface, RAS and ABL inside, and MYC in the receptor of the nucleus as well. Ret gene mutation responsible for multiple endocrine neoplasia. MET gene mutation is responsible for, this is popularly, renal carcinoma. Renal cancer is again rare, same as heart cancer. It is very rare. We do have much more breast, brain, prostate, cervix, but this one is very rare. Another one is again rare, but they are responsible for signal transduction. They are oncogene. RAS gene mutation, 3 group of classes we have: HRAS, KRAS, NRAS. All 3 together responsible for 10 to 15% of all human cancer. And you can read in the bladder cancer, colorectal, and also non-lymphocytic leukemia. These are responsible for this kind of mutation. Just a summary for the first session: cancer is the second cause of death in the developed country. Incidence and death rate vary between gender and race and ethnicity. I told you, for example, in Gombad we have the first esophageal cancer around the world, which is not the case in the other country. Or maybe we say Burkitt lymphoma is much more in Africa than in Asia. Can be defined by the cell type, as I told you, epithelial or mesenchymal. Classified by the various staging. That's another important issue. Staging means TNM. TNM. You will see it in the pathology report. T means size of the tumor. Size of tumor. N means how many nodule is involved, lymph node. Usually by CT scan MRI we say, okay, the lady has got cervix cancer and there is involvement in nodule, for example, around the aorta, nodule, and metastasis. And we say T0N1M1 or T2N1M1 or T3N2M3, for example, something like that. This is a staging. Means, is it progressing very quickly or is it progressing very slowly? It depends on the patient, different from each other. A multi-step cell proliferation and cell death imbalance. 3 major cancer genes we have. We just told you about chromatoncogene. You will hear about tumor suppression in the next one. And the kind of mutation, how they would be activated. Is it by point mutation, same as RAS? Is it by chromosomal translocation, same as BCR-ABL? This is 2 oncogene, normal one, fused together, fusion protein. It would be chromosomal translocation. Exactly the same one we have for the MYC and immunoglobulin, resulting in the Burkitt lymphoma, which is much more in the Africa than in Asia. This one is much more in Africa. This gene, when behind immunoglobulin, it results Burkitt lymphoma. Again, fusion protein, fusion gene. Also ERBB2 amplification, resulting in the breast cancer. And we do have a drug for that, herceptin would be very good one. So we usually take a biopsy from the breast of the lady. Then we do checking by the immunohistochemistry. So immunohistochemistry, for false language. Immunohistochemistry. It means painting by a specific color to see, is it positive or is it negative? Shall we go for this treatment or shall we not go for this kind of treatment? Can be done by this. Okay, 10-minute break up to